Proper wound dressing is a very crucial yet often overlooked factor in promoting healing. Improper wound dressing could lead to myriads of problems such as infections and sepsis, which are involved in 1 in 18 deaths in Canada. Frequently changing wound dressings to avoid infections cools down the wound tissue and stops healing for up to 4 hours, thus lengthening this healing process. Hence, we present ChitoZing, a kit which includes fixed amounts of chitosan embedded with 6-gingerol in powder form and a dissolving solution that, when combined, will make a gel that can be applied to any wound so that frequent dressing changes could be avoided. We are devoted to the synthesis and testing of this novel synthetic biology project so that patients in rural communities or on reserves with open wounds who have no time or means to pay visit to the hospitals for often dressing change could benefit from this solution.
Current wound dressings provide a temporary physical barrier but provide poor adherence to wounds, antimicrobials that lead to resistant bacteria, and do not store medication for long-term release. To address these shortcomings, ChitoZing aims to create a low-maintenance wound packing material that can be easily modified based on the patient's needs. The uniqueness of our product comes from the incorporation of 6-gingerol, a corn-sensing inhibitor that uses cell-to-cell communication between bacteria as the marker for the bacteria. By disrupting the communication between bacteria, 6-gingerol is able to stall the formation of harmful bacterial colonies on open wounds and trigger a quicker and stronger immune response. In addition, the value and innovation that comes with the product lies in its ability to change color based on the extent of exposure to open wound exudate. ChitoZing aims to incorporate a polymeric dye by introducing a substance such as anthocyanin or sweet potato extract that will change the color of the dressing to indicate whether it is time to change the wound dressing. By improving technical aspects of wound dressing, our project will have addressed Sustainable Development Goal 3 by promoting good health and well-being.
ChitoZing will come in the form of a kit that will include various solutions, mixing agents, and models. The ChitoZing, which will be a pre-measured amount of chitosan embedded with 6-gingerol in powder form, will combine with 2 different solutions to form either a hydrogel bandage or a wound packing paste that seals the wound and promotes isolated healing. The specific technology used, including sequencing, PCR, gene amplification, insertion, and verification using mass spectrometry, Western blot, and other similar tools, will revolve around 4 main pillars: developing the antibiotic, developing the chitosan, promoting an iterative verification process, and using parallel approaches to create a chemical working model that can be explored superficially while the genetic circuit is being designed. First, we have the antibiotic. The main concept of ChitoZing is that it will encompass a genetic linker sequence that introduces 6-gingerol to the chitosan foundation in an equivalent and dispersed manner so as to avoid toxicity. Then we have the chitosan itself. Chitosan will be purified from stress-induced yeast spores, amplification of chitin synthase, and chitin deacetylases under nutrient stress will optimize the 6-gingerol-linked chitosan in vivo. Then we have the verification and the 2-approach process. Mass spec, gel electrophoresis, and Western blots will enable the verification process. To assess the merit of the dressing, such as the efficiency and longevity, a hydrogel dermal layer will be used. While the synthetic biology circuit is being designed and tested, chitosan cross-linked with 6-gingerol chemically will be studied as a working model to offer insight as well as grounds for the symbio approach. After connecting with the community and stakeholders, it was determined that synthetic biology would be the most safe, cost-effective, and useful way to generate a wound packing material that is both versatile and equally dispersed as 6-gingerol to introduce antimicrobials and avoid patient toxicity. This was the first step in delineating the need for synthetic biology as opposed to using harmful chemical reagents that do not adequately promote chitin deacetylation and cross-linking with 6-gingerol, which results in an aggregation of the antibiotic and a potential for adverse effects. By having a working model, such as the back-end chemical synthesis, although it will not be as efficient or effective as our final biologically synthesized structure, we will be able to share it with the community and engage in human practices-oriented conversations to further our synthetic biology concept and make alterations and changes as we go along. This will make room for an iterative approach whereby stakeholders, including patients, doctors, nurses, hospital staff, field professionals, researchers, and many others, can provide us their input as we go along and can adjust our project accordingly. The main purpose of our product is to deliver a consumer-friendly and accessible topical dressing and packing option. Therefore, our prototype will contain 2 avenues to help us reach our goal optimally. First, the chemically synthesized working model of our product would provide a basic design to understand the construct and compatibility of gingerol embedded in ChitoZing and parallel the longevity and efficiency of the product to combat infection on a makeshift hydrogel dermal layer. Using common cross-linking reagents such as glutaraldehyde, we would use pre-ordered ChitoZing and mix with 6-gingerol in solution. By targeting cross-linking sites in an emulsion cross-linking method, using ChitoZing as the wall material, we will generate a series of emulsions that can be suspended and isolated and then dried into a solid form. Similarly, a ChitoZing-only control would be established as a secondary working model to characterize the material and its uses. The second avenue will be the genetic circuit design, which will adequately and equally disperse the 6-gingerol component of our product in a way that hinders gingerol toxicity and enables antibiotic properties and healing. To design the circuit, we would insert the 6-gingerol gene sequence into our expression vector and connect it to our ChitoZing sequence using a genetic linker. It is important to note that yeast naturally produce ChitoZing in a nutrient-stressed state, so we will also incorporate this metabolic pathway to take advantage of the yeast's current working system. By amplifying the chitin synthase and the chitin deacylase genes, we will encourage ChitoZing synthesis that can be isolated via lysis and centrifugation. Once we sequence the genes of interest, we will amplify these regions using ribosomal binding site amplification methods. Using various mediums, such as water, glycerin, cream, and so on, we would test the consistency and character of the product in various solutions to create either a wet-dry wound packing as well as a hydrogel topical dressing using the same powder. Finally, gel electrophoresis, Western blotting, and mass spec would help us to assess the success of the expressed polymer, while continuous connection with community members would ensure that we incorporate iterative feedback. Our prototype would then constitute two considerations. The first would be the front and back end synthesis approaches, and the second consideration would be the final kit that would be distributed. Our team experienced concerns pertinent to the customizability of our product, which we were able to pinpoint and specify after very thorough discourse with community members, including nurses, doctors, field professionals, and other scientists. We finally decided that rather than creating a hydrogel bandage, we wanted to create a more versatile product that can be applied in different manners, such as making a wound dressing hydrogel, but also having the means to create a wound packing, so that it can be altered to help with chronic and acute, superficial and deep wounds of all statures and requirements. Having the ability to mix in various other medications, such as particular antibiotics, would provide a more in-depth character to our product alongside its internal antibiotic and healing capacities. We also faced difficulties in the context of identifying associated weaknesses with the synthetic biology scopes of our product. Understanding the various gene expression parameters helped us to figure out that amplifying the inherent ChitoZing production would help with our own project, while also mitigating the limited scalability of our product. All-inclusive, the various difficulties faced by our team in the process of developing our product helped us to design and generate a final working model that is wholly iterative and community-oriented in nature.